Uh, Good afternoon, everyone, and welcome to my talk. I am Debayan Dasgupta. I work at the Center for Nanoscience from Professor Ghosh's lab. I completed my PhD last year, and this is broadly my PhD topic. But this is the outline that I'm going to talk about. I'm going to talk about how to do microrheology using nanomotors, probing the cancer microenvironment, and if time permits, I'm going to talk about startup that we co-founded, and this is a little bit of self-promotion, but this is when nanorobots are going into the real world. So before I start going into the details, a little bit about why we use this sort of geometry. It's as anyone who works in low Reynolds number physics knows that reciprocal motion like the one that scallops do, they don't work very well, or they don't work in low Reynolds number regime. You need to break the symmetry, and the way we do it is by using a geometric asymmetry, which is a helix, and we rotate this helix and they move like corkscrew through the fluid. They can translate because of the translational coupling. Our lab has been working on this for almost 10 years. My seniors have figured out the dynamics of nanomotors, and then my other seniors have applied this technology in biology. This is how we fabricate nanorobots. We use something called glancing angle deposition, where it's a shadow-based technique where your substrate is at an angle and it rotates, and that allows you to create helical shapes. This is the setup that we use. It's a very simple setup where we have a triaxial Helmholtz coil that is run by 3 amplifiers and is controlled by a computer. And we also simultaneously record what's going on using a microscope. So now coming to microrheology using nanomotors, I'm going to talk about why it's useful, why do we want to do this, and what sort of new information that we can get using nanomotors as rheometers. So there are a lot of microrheology techniques in the world, and some of them are highlighted over here. They are pretty good on their own, but they are not very fast. They can't measure local heterogeneity as well as we would like to, and they cannot create a spatiotemporal map of viscosity, which is what we really need to go into cancer. And the way this happens, let me see if I can run the video. Yeah. So if you go through the dynamics of nanomotors, if you solve those equations, you will get to a regime of motion, actually 3. Over here, we are bothered about 2 regimes of motion. One where the nanomotor perfectly follows the applied magnetic field. There's a rotating magnetic field, and it tumbles. And that is before a certain cutoff frequency. But as you increase the rotating frequency, it starts to precess, and this is what we call precessional motion. Whenever there is precession, we can measure the angle of precession, and that is the part over here. This is alpha p. This is the angle of precession, and it follows a sine inverse curve, as you can see over here. And the sine inverse depends on multiple parameters, and we know all of them. One is the magnetic moment that our nanomotors have. B is the applied magnetic field. Theta m is the angle between the magnetic moment and the body normal. 2 pi fs is a geometric drag coefficient, is a geometric factor. Eta is the viscosity of the fluid, and omega b is the actuation frequency that we plot over here. So if you rearrange this equation, you can get out the viscosity term from this equation, and that is how we can measure viscosity using nanomotors. So we took this and we tried in multiple fluids. Some of them are Newtonian, which is glycerol and water, and we don't see any shear thinning property. But if you go to a shear thinning fluid like xanthan gum, you can see that as we increase the frequency, the actuation frequency, we can see the shear thinning property. So this proves that we are measuring shear thinning and we are measuring viscosity using nanomotors. This allows us to, because this is very fast, this is almost 100 times faster than current technology. This allows us to measure viscosity in real time. And this is the cooling water. So the water was heated up to 70 degrees and we allowed it to cool to room temperature, and water's viscosity changes from 0.4 to about 0.8, which is room temperature. Now over here, the stars are the data that we got from passive rheology, and you can see the amount of error that is there in time, because you need long data to get temperature from, to get any information from passive rheology. Our information is much quicker. You have an error bar here, but you can't see it because it's much better than passive rheology. We can also do some lot of fun stuff, which is we can move from water to glycerol in a mixing fluid, and we can bypass the mixing time because this is so fast. And therefore, we started at water, we drove across the microfluidic chamber, and we reached glycerol, which was 62 cp. And this is a proof that we reached into pure glycerol from water. So with all this, the reason for doing all this physics is because we wanted to apply into biofluids, and we chose two important biofluids. One of them is gelatin, the other one is matrigel, and you can see that shear thinning property can be seen from matrigel. And this is where I'll go to the next point, which is we use matrigel and cancer cells. So this is the takeaway from this part of the talk, is that you can measure non-Newtonian and Newtonian properties with high spatiotemporal accuracy. So now I want to look into cancer microenvironment. We want to see if we can maneuver in a tumor-like environment, in a tumor model. So this is an engineer's view of cancer and cancer microenvironment. I'm not a biologist, I'm an engineer. And the way I understand it is that normal cells have this well-aligned patterns. They are sitting on a basement membrane. One of those cells become cancerous and they start invading the tissue. And the way they do it is they release matrix metalloproteinase that cuts into the normal tissue, and they secrete their own ECM. Now, ECM is extracellular matrix. It has a lot of proteoglycans, fibrous proteins, and it's used by the cells to communicate among themselves. And we had to use a 3D structure, a 3D cell model. This was all done by Professor Ramre's lab in IISc. And to use this matrix, we use matrigel matrix. And we need 3D because in 2D there are a lot of other artifacts that we don't want. So the cell has to feel at home in a 3D environment. And when we do that, what we find is if we drive nanorobots in this matrix, it can swim past normal cells, but whenever it comes close to a cancer cell, it gets stuck. So there is no adhesion to a normal cell, but they eventually adhere to the normal cell surface maybe, but they will always get stuck at a distance from the cancer cell. And this adhesion was observed up to 40 microns from the cell surface. And I don't know if the video is running. Yeah. Yeah. So on that side, this is a cancer cells, and over here you will be seeing normal cells. This had to be sped up because this is a much longer video, and you can see that this guy just comes and sticks to the cancer cell surface, not the surface, a little bit above, far from the cancer cell. But this guy keeps on poking the normal cell for a long time. If it gets stuck, it will eventually get stuck on the surface, not in the microenvironment. And the reason for this is cells secrete a lot of proteins into their ECM. This is again a very simplified explanation. There are a lot of complex biology that goes on here, but what you need to worry about over here are the sialic acids, and cancer cells are known to be hypersialylated. So they secrete the sialic acids so some of the sialic acids are on the cell surface and some of them are secreted into the extracellular matrix. And when we stained for sialic acid and we stained for the cell surface, what we found is that this 231 is a cancer cell and HMM is a normal cell. So what we found is that it secretes ECM it secretes sialic acid far into the ECM. So what it indicates is that it's eating away the native ECM and it's secreting its own ECM. And sialic acid is a very charged molecule, and that's why it's sticking, but I'm coming to that. The other fun thing is there is a cell-to-cell variability. So 231 type cells release a symmetric form of sialic acid, and this is also seen with the way the nanomotors stick to the cancer cell. For MCF7, this the motion, the way sialic acid is left from the MCF7 cells, again it's anisotropic, and our nanomotors also seem to be seeing the same thing. They get stuck in an anisotropic fashion. So I don't know if the video is running, sorry. Yeah, so this was the hypothesis that the charge of the sialic acid is what's causing these guys to stick near the cancer cell, and therefore we did a control experiment where one of the nanomotors was not coated and the other guy was coated with something called PFO, which is a charge shielding molecule, and you can see that the charge shielded nanomotor keeps on probing the cancer cell vicinity, whereas the normal the uncoated nanomotors is always stuck. So this proves that sialic acid and the charge is causing all the problem. Now, what are the applications of this finding? One of them is that you can use it for cancer targeting. So we did some simulations and we saw that if you have such a boundary around a cancer cell, they will eventually get stuck, and if you keep doing this nanomotors back and forth motion, if you send them and then you bring them back, they will eventually localize around the cancer cells, leaving the normal cells as it is. And this is a very neat way of cancer targeting. The other application is something that we can gauge the metastatic potential. So the number of cells that we use, the cell lines that we use, they have different metastatic potential. 231 and MCF7 are supposed to be very aggressive cancers, and you can see that the force needed to reach the cells, the force needed to overcome this adhesion and move, is much higher than the force that is needed by these two. These are rather tame cancer versions. And then we have normal cells where there is no adhesion, and therefore the force is calculated from the velocity. Yeah. So also the other application is something that we discovered new phenomena. One of them is the anisotropic distribution of cellulation, and this is something that the biologists are very excited about. So since I have 1 more minute, I'll be talking about where these nanomotors go into real world, and this is something fascinating. This is about dentistry and something that we never thought about. So when you go for a root canal treatment, your root gets infected and the doctor removes the pulp, and there is something called dentine tissue over here. So when that happens, dentines have this small holes and long tubes over here. And what happens is, due to improper cleaning, and this is not the doctor's fault, due to improper cleaning, there are bacteria in these dentinal tubules. And this is E. faecalis. They don't cause too much problem, but they stay there. But eventually, when your immunity is shot, they will cause a lot of trouble and they will cause root canal failure. And modern technology cannot solve this problem, because you will see that diffusion and everything is very bad. But nanomotors can. And what we did is we drove nanomotors into the dentinal tubules. We drove them deep inside, and this is already better than what current technology can do. This is a confocal image of a cross section where these are all nanomotors. And once this is done, we can bring them back. We can heat them up using hypothermia. So that's it. Thank you.
Thank you for the excellent talk. We have time for 2 or 3 questions. Is there a question from the audience? There is a microphone right next to you.
Yeah, that was a fantastic talk. So,
Thank you.
I had a doubt regarding the nanomotors localization method.
Yeah.
So particularly with regard to the cancer cells.
Yes.
So you're relying on a differential surface charge.
Yes.
In order to migrate it towards a chosen locale.
No, the migration is happening because of the magnetic field that we generate. It's a
that depends on the surface charges.
No, this is no. This is a magnetic field. So if you go back to the first slide, I think the way this moves is, so the nanomotors have a magnetic moment on them. So they have a magnetic material in them. So they have a magnetic moment, and when this coil generates a rotating field, it follows that magnetic field. So it's like a corkscrew that keeps on moving. So it moves like that. So the motion of nanomotor does not rely on the surface charge or anything. It gets adhered because of the charge, because the cancer has secreted charged ECM.
Okay, so the way you distinguish between a regular cell and a cancerous cell is based on the difference in the surface charge.
Yes.
So in that particular, so the experiment which you have shown us, it's in an in vitro cell culture, is it?
Yes.
So have you tried this in vivo, in a mouse model?
Yeah. So there are people in the lab who are trying it right now, because the amount of field needed to move in a live animal is much higher. This can generate only 200 Gauss. Now we have huge coils which can generate almost 1000 Gauss or even more. And that will test whether this works in a mouse model.
Because in that particular instance, you will have a wide variety of sublocales inside a living organism.
Yes.
All of it will have a differential charge expression.
Yes.
So how will you precisely maneuver the motor only to the tumor? You will need to go through all the other microenvironments.
Yeah. So we tried moving it in a breast tissue. The problem is breast tissue has a lot of adipose cells, fat cells. So it gets stuck in the fat cells. So whenever there is a phase difference, it gets stuck. So the idea is you inject the nanomotors very close to the tumor, not exactly at the tumor, but very close to the tumor. And then you keep on moving them back and forth.
Understood. Thank you.
So maybe, is there any other question? If not, please thank the speaker.